before I start with this little. Okay, so being tall or the opposite, right? Being short. Those are characteristics, definitely, right? Or small, right? So depending on whether you're talking about like a person or a plant, right? It could be like a tall plant versus a small plant. Those are traits or characteristics, okay? Very good. So those traits or characteristics help us. Are you in Miss Garza's third block? Miss Garza's third block? Yes. So those traits or characteristics help us identify the way we are and make us who we are, of course, right? So those traits or characteristics are in our chromosomes. Remember that chromosomes, guys, are those things that look like X's. Okay, they look like little X's. And in those X's or those chromosomes, we have DNA that's very wound up or kind of like jam packed into the chromosome. And within that DNA, we have our traits that tells us, okay, you're going to be tall. You're going to, let's say, have curly hair. You're going to have brown hair, right? You're going to have all those different types of traits or characteristics, okay? So those traits or characteristics, we actually use something to measure them or kind of try to like figure out what kind of traits or characteristics our children or offspring are going to have, okay? So this little chart that I'm showing you, it's called a monohybrid cross. So this little trait, this little cross or Punnett square, they call it that way. This is a Punnett square. This is going to show me a más o menos on how the couple here, for example, this guy and this girl, what kind of children they're going to have depending on their traits, right? So that's what monohybrid cross is, and this lesson is talking about. It's all about traits and how they are passed down from mom and dad to their offspring or their children. So I'm just going to go ahead and start presenting this little video here, and I'm going to be pausing in some parts that I think are important to pause on, just to emphasize or make sure that you guys understand what the presentation is talking about. So I'm going to go ahead and mute myself here on the Google Meet, and then I'm going to go ahead and start it, okay?
A Punnett square can be used to predict the probability of certain genotypes and phenotypes of an organism's offspring. Probability is the likelihood that an event will happen.
The genotype is the actual makeup of an organism, and each organism has two alleles for the trait. If both alleles are the same, then genotype is homozygous. If the organism has both copies of the dominant allele, then the genotype is homozygous dominant. If the organism has both copies of the recessive allele, then the genotype is homozygous recessive. When the two alleles are different, the genotype is heterozygous. The phenotype is the observable trait. Due to the principle of dominance, organisms having the dominant allele will express the dominant trait. So both homozygous dominant and heterozygous genotypes will express the same dominant phenotype. An organism that is homozygous recessive will express the recessive phenotype.
Punnett squares are used to predict genetic crosses. Before setting up a Punnett square, the parent's gametes must be determined. Alleles from the parent's gametes will determine the organism's genotype and phenotype.
All right, I'm going to pause here really quick just to explain to you what they're talking about, okay? Okay. Yeah, guys, can you not use that door? Can you please use the other one when you're coming in or out? Okay? It's locked? Okay, then just leave it open. All right? Or also put the little magnet down. Yeah, there you go. Okay, so really quick here on the notes that I paused here, they're talking about the parent's gametes. Okay, remember gametes are sex cells. And from what we've learned so far, we've learned that gametes for girls are the sex cells for girls are eggs. If you don't remember that, right? So the sex cell for girls or females are eggs. The sex cells or gametes for males are sperms. Now, here we don't see any pictures of eggs or sperm, obviously, right? So the gametes or the parents' gametes that they're talking about here are this one is one of them, and this is another one right here. Okay? And they represent them in letters. Okay? So these are those traits that I was talking about to you guys. This is parent number one. Let's say this is the mom right here in yellow. And then this one over here is the dad. Okay? Or it can be the other way around. It really doesn't matter the order of which where goes who goes where, in other words, okay? But these are the parents' gametes. Another name they might give these letters are genotypes. Okay, so we can also say that these letters at the top and the left side here of the Punnett square also represent the parents' genotypes. Genotypes, as in their letters. Those letters, what are they there for? They're representing again the trait. The trait that they're measuring here for is the pea pod color. You see that? This one is yellow, this pea pod is yellow here, and this pea pod over here is green. So this big Y and little y represent a yellow pea pod for this parent. This one on the top, the two little y's here represent a green pea pod. Now, there's something else to notice as well. You're probably wondering, well, why does it have two letters for each parent? It has two letters per parent because you're going to get half of the traits from mom and the other half from dad. It's kind of like when you were talking about chromosomes and meiosis when you were covering that with Miss Garza. Remember, you get 23 chromosomes from mom, 23 chromosomes from dad. You always get half from mom, half from dad, half and half, right? And then it makes you who you are. Sometimes we look a little bit more like mom, and then sometimes we look a little bit more like dad. Sometimes we're like a little mix of the two, or sometimes we might look like more like our tia or our tío or grandma or grandpa, right? Sometimes those looks or those traits, they skip a generation. So it just depends, right, on what type of uh, mixture of mom and dad's DNA you get and which one is more presentable or stronger from what you can see on your features, okay? So that's why each parent here has two letters, a little bit of mom, a little bit of dad, right? So now their parents, right here in this Punnett square here. So this is one parent here. That's what I'm talking about, right? And this is another one over here. Okay, mom over here on the side, dad over here. Now here in the middle, these are the four different possibilities of how their babies or the baby pea pods, right, are gonna look like. So how do they figure that out? They cross their letters. For example, this big Y, they placed it over here. You see that? This little y, they placed it down here. So that's why I have a big Y, little y. And it's going to look just like mom. And it's going to have a yellow pea pod right here, just like mom. And the same letters. You see that? Or same genotype. So whenever we're placing the letters, guys, we put the capital letter first, and then we put the lowercase one right next to it. Okay? Now, let's check. Let me erase all this because I don't want to eat it. Now, let's check this other one. Big Y is going to come over here from mom, right? Little y down here from dad. You see that? So it gets a little bit of mom, a little bit of dad. That's why we have two letters per pea pod, right? Or individual. Same thing over here in the bottom. This little y comes from mom, and then this little y comes from dad. You see that? Same thing for this last one. This little y again is going to come over here first. This little y is going to come from dad over here right next to it. So that's how you cross a monohybrid cross and find out how the offspring are going to look like. Now, there's something else important that you need to know about this. each little square is worth 25%. Okay? So if each one is worth 25%, how much is that in total? 25 times 4? How much is that? Or you can add each one individually. If they're 25 each, how much is that in total? Okay, so one square is 25%, right? If the other one is 25 as well, and this one is also 25, right? And also this one is 25, how much is that in total for all four? So 25 and 25 is 50, these two on the top, right? And then another 50, how much is that? 100, there you go, okay? So this is 50 right here for these two, right? And this is another 50 right here. So 50 plus 50, 100. So everything is supposed to add up to 100% as far as the numbers or percentage-wise for my Punnett square, okay? So again, they're going to ask you this on the handout we're going to do right now for class click in a little bit. They're going to ask you, what do the outside genotypes or letters represent? You're going to say parents, right? And then they're going to ask you also, what do the genotypes or letters on the inside represent? You're going to say offspring or children. You can put that as well. And then another name for alleles, guys, are genes, as in genotypes, okay? Just so that you won't get confused on that vocabulary term. All right, so I'm just going to go ahead and move on. And then we're going to do one more example, and then we'll work on the class cake assignment. Okay, I'm going to mute myself here on Google Meet.
During meiosis, which forms the sex cells, copies of a gene separate so that each gamete will receive one allele or the other. This is called the law of segregation. To predict a genetic cross, the possible alleles from one parent's gametes are placed along the top of the Punnett square, and the possible alleles of the other parent are placed down the side of the Punnett square. After alleles are in place, the squares inside the diagram are filled in with the appropriate paired alleles. In this example, the gene for a widow's peak hairline is dominant big W. The gene for no widow's peak is recessive little w. One parent is heterozygous for widow's peak, big W, little w. The other parent is homozygous recessive for no widow's peak, little w, little w.
Okay, we're going to go over this setup that they have here for us, and we're going to work it out together, okay? But first, let me explain those new terms that they have for you there, just so to make sure that you understand what they're putting there for you, okay? So, a little bit smaller, there we go. So the data up here, right? They're saying his letters are big W, little w. But look at this. This is part of your vocabulary. If you don't remember it, heterozygous means I'm going to have two different letters. Two different letters as in one's going to be capital, like this one, and the other one is going to be lowercase. That's what they mean by heterozygous has different letters. Technically, they're both W's, right? But one of them is a capitalized one, and the other one is a lowercase one. That's why we call them different letters, okay? So dad is heterozygous for widow's peak, meaning he's going to have that dominant big W that gives him his widow peak, or the little point that's right here in his forehead. Like me, I don't have widow's peak, right? So I would be like the lady there. I would have two little W's in this case, right? Because she has no widow's peak. So since me, I don't have widow's peak, right? And if you're like me, that you don't have a widow's peak here on your forehead, you would be homozygous recessive, like the mom or this lady here. So you would have that recessive gene or letter there that stands for no widow's peak. But if you do have that little piquito here on your forehead, You're probably like this guy, and you're gonna have that big W, which is the dominant trait, which stands for widow's peak, or means you're gonna have that little piquito for widow's peak, like this guy right here. Okay, so these are just the parents. Okay, heterozygous again, I'm gonna have two different letters, one capital, one lowercase. Does it have to be W's? No. It could be any letter from the alphabet. It just depends on the word problem or the example that they give you guys, okay? Don't be like, oh, okay, she said or she used only W's, it's only going to be the letter W. That's not true, okay? It can be any letter of the alphabet. This is just an example, okay? Just wanted to make that clear. And then homozygous recessive is when I have two little letters, like these two little W's. So here, they already have the Punnett square or my monohybrid cross already set up. So we're only going to be measuring one trait. That one trait is whether the individual is going to have a widow's peak or not, right? That's my trait that we're measuring for, widow's peak in this example. So here I'm just going to write down my letters. I'm going to put my big W here. This is that big W that I'm getting from. And then this little W, I'm going to put it right here next to it. So these are dad's letters. I'm just setting them up again here so that we don't get confused when we're crossing the Punnett square. And then mom has two little W's. So I'm going to put one little W of hers right here and the other one right here. Okay? Don't pay attention to this right now. So we're going to cross them. For this first square, remember it's for 25%, right? So that means this individual, if they have a baby that's going to come out like this, it's going to be a 25% chance that this couple has the baby like that. So hold on. I have, let's see. It's going to be, yes, Jerry, you're right, but it's going to be the other way around, Jerry. You're going to put there you go. Capital letter first and then the lowercase. Okay, no problem. So here, let's put big W, little w, right? I like to put the capital letter first since it's the dominant one, right? Anytime we have a capital letter, it's going to be a dominant trait. So it's going to go first. What about this other one over here? The second square. What are we going to put here? Okay, look, I have a little W here. There we go, Leslie, right? His little W is going to go down here. And then mom's little W is going to go right next to it. Sorry if you can't really see the way the W's are. I tried to make them big and small. With W's, it's a little bit hard to distinguish which one's big and small. There we go. I hope that helps. What about this other one in the bottom? Very good. So that is big W from dad, right? It's coming down here. And then mom's little W is going right next to it, right here. Okay? So the thing is, they're all W's. I understand that. I know why you put two W's, Leslie. But I like to tell you big or small since I have capital ones and lowercase ones. In this case, it matters whether you say or you write it down as a capital letter or a lowercase letter. Okay? So for here, it does count, even though they're all technically W's. Okay? What about this last one right here? What are we going to write down here for this one? So we're going to get dad's little W and put it down here. And then mom's little W and put it right here. Right? So, and remember, each one is worth 25%. Now, there's a pattern here. Look at the ones here on the left. Both of them have a big W, little w. See that? Big W, little w. So these are technically two individuals that are going to have widow's peak just like dad because they have dad's letters. You see that? So that means this couple has a 50, right? 25 plus 25 is 50. They have a 50% chance of having a child with widow's peak, just like that. Okay, so that covers this part right here. Look at the right side over here. They both have little W's. You see that? Both little W's. So here, this couple has another 50% chance, but of having kids with no widow's peak, like mom. So they have a 50-50 chance of having either kids with widow's peak or no widow's peak. Okay? All right, I'm gonna go ahead and erase this. I think this is one of the last things. Right now, they're just gonna show you how to work it out, and it'll just kind of re-emphasize what I just taught you right now.
The possible genotypes for their offspring are big W and little w in a 2:2 ratio. The possible phenotypes for their offspring are also in a 2:2 ratio.
All right, that's it, right? All they showed you was how to figure it out. Basically what I just showed you right now. Okay, so now we're gonna work on the class kick here. It's under week 13 assignments, but let me go ahead and give that to you really quick here. Or you can open it up on your own if you have it. Or where you know where it is. Copy. There you go. Start sharing this. All right, make sure you do open up the class kit because I'm going to be covering the answers. And if, of course, if you don't follow along, well, then you won't get the answers, right? So make sure you follow along. Your earpods? Just make sure you put it away, please. Okay? Hold on. And make sure you're following along as well, please, and not doing other things. All right, part A, vocabulary first. Don't do that, sir, please. Okay? Number 1, genotype made of the same alleles. What? Yep, that's what I just said. Okay, number 1, genotype made of the same alleles. What do you think that answer is? So the one with the same letters. What letter do you think that is, guys? Okay, someone said C and someone said A. Both of you are incorrect, unfortunately. Okay, so which one? B? No. D. D as in dog. So that number 1 is D as in dog. Look at the little tools at the top that you have for class kids. Just put it right there next to it. Okay, number 2, different forms of genes for a single trait. A. Lucky guess. So that one is A. All right, number 3. Genes that are always expressed. Nope. E as in it, no, not E as in elephant. That was not correct. B as in baby is correct for number 3. So gene that is expressed only in the homozygous state. Number 4. No. E is the correct answer for number 4. E as in elephant. So then that leaves us just with C, right? Heterozygous. Phenotypes made out of different alleles or 2 different letters, like big W, little w. So make sure you type them out or write them out on ClassKick. You have the little tools there on the top for you to use. Okay. Below each of the following words or choices, circle the choices that you think are correct, of course, right? So let me just, hold on. Let me use the circle option here. Okay, so number 6 is asking for the dominant alleles. In other words, circle the capitalized ones. So what am I going to circle here? Okay, very good, Leslie. So I'm going to first circle the big D there, right here at the beginning. Right there. And then what else am I going to circle? L, very good, or big L. I'm going to circle the big R as well. The N as well, you just missed that one right here, right? Right here in the middle. And the S, right? Right here, the end. Okay. So all the other ones are small. And I said only capital, right? So that's it for number 6. So number 7 is asking for recessive. So recessive are going to be the lowercase letters. So N is going to be the first one to circle. Very good. D, right? R. And K. Right here. Okay, number 8. Okay, so it's asking for homozygous dominant. So that means they're going to be the same letter and they're going to be capitalized. So what am I going to start to circle here for 8? Same letter, capitalized. So big A, big K right here, and big T. Make sure that you emphasize or you say whether it's small or big, okay? What's going on with the shield, guys? Please leave the shields alone, okay? All right. You break it, you buy it, all right? I don't know, but it was a lot. Yes. All right, so homozygous recessive, this is opposite of 8. So what am I going to circle? Sure, please don't. Leave it alone. Thank you. All right, so if it's opposite of number 8, what am I going to start to circle here? Opposite of 8. The E's an elephant, right? This is one of them. What else? Okay, the O's, actually it has a capital O and a little o. So not that one. The W's, yes, very good. And the U's, it looks like it, right? But it actually has a big U and a little u. Okay, so not that one. They both have to be small letters. Oh, I heard him say U. U. But the Q's, yes. I heard U. U is an umbrella. I didn't hear Q. So Q's are included here as well. Yes. And that's it for that one. So if it's dominant, is it going to be capitalized or lowercase? Okay. So which ones am I going to circle here? So hold on. Big A first. What else? Anything else that starts with a capital letter? The E's, the J, big J, little j, right? Even though look, yes. And the reason being is, look, because it's saying the dominant gene must show as long as it starts with a capital letter, even though it has a little letter next to it, the dominant gene is going to show still. Because look, the capital letter or the big letter is going to overpower the little one, always. Okay? So even though that little letter is right next to the capital letter, the dominant gene or the capital letter is going to overpower the little letter. Okay? So we're also going to circle this one too. Big D, little d as well. And then we're missing the big R, and the S's. Very good. Okay? So the only one we're not going to circle are the little f. So then 11 is opposite of what they were asking us in 10. So it's the little a. Whoops, I circled that wrong. And the little r. And that's it. Everybody else is dominant. Okay, so just take some time there to put all your answers there on class kits. Make sure you're circling them so that Ms. Garza marks them correct. Okay, number 12. Examine the following Punnett squares and circle the ones that are correct. So they already did them for us. We need to check them. And the ones that are completely correct, we circle them. That's what they want us to do. So this first one, I have parent number 1 on the top, parent number 2 over here on the left. Let's check each little square inside to see if they crossed them correctly. So big D, little d, little d. This one is correct. So hold on, let me put a little check mark here. Hold on. Okay. So this one is correct. I'm gonna put a little check mark right here. Check. This one, little d, little d. So that one's also correct. You okay? You all right? Okay, so this one's also correct. This next one, big D, little d, that one's also good, correct. What about this last one? Correct as well, little d, little d. So I'm gonna circle the whole thing. So let me get my circle here and circle the whole thing, everything, all this right here. Okay. Now let's check the other one right next to it. Big D, little d. This one is correct. Okay. Let's take this one right here. Big D, supposed to be a little d over here, but they put a big D. So this one is wrong right here. So if this one's wrong, The whole thing's wrong, and we don't circle it. We skip to the next one. So the next one, big A, little a, this one is right or correct. Next one, I have a little a. That one's wrong because it's supposed to have a big A, right? So if this one's wrong, the whole thing's wrong. And we skip to the last one. Next, big A, little a. Yes, this one's good. Check. Little a, correct. Big A, correct. And little a, correct. So the whole thing's right, and I circle the whole thing. There we go. All right, now let's answer 13. So 13 asks, what do the letters on the outside of the Punnett square stand for? So the ones on the top and the left, who are those? We had covered it on the little video presentation that I went over right now. So like the ones on the outside, the letters that are on the top and the left, who are those? Those letters represent who? Like, for example, the one that I have circled there, this big D, little d, that's on the top, and these two little d's, these are who? Who do they represent? Yeah. I'm just using part of 12 to answer 13. Yeah. So on the outside of the Punnett square, the letters that are on the top and the left. Very good. Vanessa and Jerry got it correct. They're the parents. So here we can put it represents the parents, and I'm just going to add the word genotype. Genotypes. Very good. Good job. And then number 14, hold on. Is asking for the ones on the inside. Who is the inside letters represent? The kids, right? The offspring. Very good. So we can put here, it represents the kids or offspring genotype. And I put genotype because look, they're representing them in letters. They're not putting like boy or girl, obviously, right? So that's why I put the word genotype, because they're representing them in letters. Okay. Oops. There we go. So once we start on 15, that's your second slide on Class Kick, okay? So once I scroll down to number 15, that's your second slide on Class Kick. All right, number 15. It says in corn plants, normal height or big N is dominant to short height, little n. So the dominant letter or the capital one that we have here is N, big N, okay? And then short height, as in like a short corn plant, is represented by a little n. So remember, that's going to be your recessive letter. So I'm just going to add here the word recessive. You don't have to add it. It's something extra, okay? Complete the 4 Punnett squares showing different crosses. We're gonna shade in all the homozygous dominant ones red. Right now we'll work on that. Shade in all the heterozygous ones green, and then shade in all the homozygous recessive ones Unshaded. Leave it white. Okay? So they already set up everything. All we have to do is cross them. So what am I going to type in here? What am I going to start off here, guys? What two letters am I going to cross with here? Look at the one on the top and then the other one on the side. Very good, Jerry. Good job. So it's going to be big N, little n. Okay. What about the one next to it? Follow the same thing, right? The top one and then the side one. And then you put them together, right? Top, side. So that's also big N, little n, right? Go to the bottom here. Big N, little n. Again, I'm getting the big N here from parent number 1 on the top. And then moving over little n from parent number 2 over here from the side. Okay? So here, big N, little n. All right, so since they're all big N, little n, and they're different letters, what color am I going to shade them in? Look at the parts that are underlined in the problem. What color? There's no blue, so we're going to shade them in green. Okay? So green is heterozygous. Heterozygous means I'm going to have two different letters, and I do have two different letters. I have a capital N and a lowercase n all throughout this square. So they're all going to be green. So here, hold on. There's my, oh, not that one. Green. Okay, so all highlighted green. I don't know if you have this option on Classcake or not, guys, but if you do, you can color it in or shade it in green, okay? Okay. I think you have the option of changing the pen color, I think. You can just like outline it green if you want, okay? All right, so now let's do the next one. What's gonna go right here? What two letters? Big N, very good, Jerry. What else? Right here. What do I put here? Very good. Big N, small n. Okay, and the bottom one? Two big N's. Very good. And the last one? Yes, big N, small n. Good job. Now, since in the other one, we said the big N, small n's go green, we can color the ones on the right here green, right? Because they're all big and little n. So let's shade them in green for those that we know go green, which is the right side of the square. What about the other side, the ones with the big N's? What color do those go? Look at the little paragraph on the top. So these are heterozygous. These that are capital, are they dominant or recessive, the capital letters? Okay, so remember, recessive are the little letters. Okay, those are the little letters. So the dominant ones then would be the capital letters. And we both have the same letter, both big N's there for the left side, right? So they're both big N's as they're going to be homozygous. And we said they're dominant because they're capital letters. So they're going to be homozygous, dominant. So that means they're going to be red. So those go red here on the left side of this second one. red. Okay, now let's do this other one. What goes right here? Two big ends, very good. What else? Right here in the side. Big and small end, correct. What else? What else? It's those letters, but the other way around. Big end first, and then small end. Okay? Always put the big letter first. Okay? And then the last one, two small ends. Very good. All right. How many squares am I gonna shade in green here for this one? Two. Very good. So those are the big and small ends, right? So let's come out with the little highlighter that I have here. They're gonna be diagonal from one another, or right across from one. Oh, I had it at red. My bad. I have to shade it in it. There. Green for that one, and then green for this one. Any reds? Yes, right? The big ends. Right here. So this one goes red because it's homozygous dominant. Does this one go any color, or do we leave it like that? We leave it like that because it's homozygous recessive. Two little ends. Recessive is when I have, for example, two little ends or two lowercase letters. Okay. Now let's just do the last one here. What do I type in here? Big and small and, oops. Okay, what about the next one? Two small ends. Very good. Right here. And the last one. Two small ends, or small end, small end. Good job. And now just to shade them in. So the greens, or the big and small ends, I have two on the left side. So shade these in green. And my homozygous recessive, or my little ends, they go unshaded. So that's it for 15. So make sure that you have all this information down on your class cake assignment, or else you won't get credit for it, okay? Slide this down a little bit. All right, so 16, it looks like we're only gonna have time for letter A, but as long as we do one example, hopefully you'll get to do, or you can do letter B on your own, okay? So in guinea pigs, short hair, big S, is dominant to long hair, little s. Complete the following Punnett squares according to the directions given. Fill in the blanks beside each Punnett square with the correct numbers. Okay. So for letter A, let me just switch to the text box here. Instead of setting them up on the Punnett square, we have to put the numbers, or sorry, the letters there. So it's telling me the parents are big S, little s, and the other one is little s, little s. So I'm going to put parent number 1 here. Big S, little s, that's one parent. Remember, they all have 2 letters. And the other one is 2 little s's. Make sure you're following along. Okay, so now I just cross them, right? What do I put here? So there you go. Big S, little s, good job. Okay, next, right here. 2 little s's, or 2 small s's. Okay, and then here. Big S, small s. And then, yes, 2 little s's, very good. So here we don't have to shade in anything, Okay. The extra step that they want us to do here, you see here where it says expected number of offspring? They're asking how many short hair, or how many do I have of big S, or big S, small s? How many do I have of those for short hair? Two. Very good. On the left side of the square, right? How many do I have for long hair? Yep. Okay. And then two again, right? Because on the right side. And that's it for A. So if you're going to try to do B on your own, follow the same steps. Okay? The only thing that I'm going to give you here that you might not understand, right here is saying that they're heterozygous. That means that both parents are going to be big S, little s for those. So if you're going to try that on your own, you're going to set up both parents with big S, little s for both of them, both on the top and the side for both of the same letters. All right, guys. So I'm going to end here because we have 2 minutes of class left. Any questions, guys? Online? Questions? All right, guys. Have a good weekend, and Ms. Garza will be back Monday. Have a great day.